Welcome, researchers, scientists, and knowledge seekers. This presentation introduces a structured, systems-level framework for understanding chemistry as a foundational driver of biological function, disease mechanisms, and therapeutic development. This framework is part of the ongoing research and systems architecture work developed by Brian Geniuson, research scientist. Chemistry operates through four core principles: molecular identity, structure, properties, and reactivity. Molecular identity defines what a compound is. Structure determines how it is arranged in three-dimensional space. Properties describe how it behaves under different conditions. Reactivity governs how it interacts and transforms. Together, these principles form the foundation of chemical transformation. As chemical transformations occur, they extend beyond isolated reactions and begin to interface with biological systems. This transition represents the biological interface. At the biological interface, molecules interact with proteins, enzymes, membranes, and signaling pathways. These interactions regulate cellular behavior through mechanisms such as molecular binding, enzymatic catalysis, signal transduction, and metabolic control. From a systems biology perspective, these processes can be mapped across interconnected molecular networks involving gene expression, protein interactions, and metabolic pathways. These interactions scale into cellular and tissue-level effects. Cells respond to chemical stimuli through adaptation, stress responses, and regulatory signaling. These responses propagate across tissues and organ systems, influencing physiological processes such as metabolism, immune function, and neural communication. When dysregulation occurs, these processes contribute to pathophysiology and disease. Disruptions in chemical balance, structural integrity, or signaling pathways can lead to complex conditions, including metabolic disorders, inflammatory diseases, neurodegeneration, and cancer. Understanding this progression enables the translation of chemistry into therapeutic development. By targeting molecular pathways and biological systems, chemistry becomes the foundation for drug discovery, formulation, and clinical intervention. This translation is supported by integrated scientific systems. Pathology mapping connects chemical processes to disease mechanisms and progression. Pharmacogenomics explains how genetic variation influences individual responses to chemical compounds. Biodistribution determines how substances are absorbed, distributed, metabolized, and excreted across tissues. Systems biology integrates these layers by mapping interactions across proteins, pathways, and cellular systems, enabling a network-level understanding of chemical influence. From a multi-omics perspective, this framework incorporates genomics, proteomics, metabolomics, and epigenomics, providing a comprehensive view of how chemical signals propagate through biological systems. Evidence supporting this framework is derived from both mechanistic and clinical domains. Mechanistic evidence includes molecular and cellular studies that define pathway interactions and biochemical processes. Clinical relevance is supported by translational and population-level research, linking chemical exposure and therapeutic intervention to measurable health outcomes. However, it is important to recognize translational constraints. Chemical effects are influenced by factors such as bioavailability, dosing, metabolism, tissue specificity, and individual variability. These factors can impact how mechanistic findings translate into real-world clinical outcomes. This highlights the importance of integrating systems-level data to improve predictive accuracy and therapeutic effectiveness. This framework provides a unified model that connects chemistry to biology, disease mechanisms, and therapeutic outcomes. It supports scalable research, clinical translation, and interdisciplinary integration across modern biomedical science. If you find this type of structured scientific content valuable, consider subscribing to follow future research and systems-level analysis. Additional extended research, visual frameworks, and full literature synthesis are available upon request. Thank you for engaging with this framework.